So thank you all very much for attending this webinar, where I will discuss Takara Bio's solution for cancer genomics and putting the accent on how our solutions can benefit your cancer genomics research, mostly on accuracy, sensitivity, and reproducibility for CNV and SNV analysis. Okay, so as you know, there are many different ways of looking at genomic alteration in cancer. Of course, the most common ones are looking at genetic variation on DNA, and this includes copy number variations, CNV, structural variants like insertions, translocations, and deletions, but also single nucleotide variants, point mutations called SNVs. So on top of this, you most probably are familiar with additional layers of investigation, like for example, epigenetic variation. So here looking at chromatin and the epigenome landscape, but as well at transcriptome of cancer cells by RNA-seq, either in bulk population or single cell analysis, and all put together. You can have, of course, a multi-omic approach, putting all these layers together, which ultimately leads, hopefully, to search for biomarkers, monitoring of disease and therapy, and nowadays mainly focusing on circulating cells, circulating tumor cells, and circulating nucleic acids. So this is a very schematic view of what one can do by looking at copy number alterations. So here is depicted a chromosome where at the bottom of the graph in blue you see copy numbers of regions of this chromosome, and for example, you can see that this one is higher than the previous one. This one is lower, etc., etc. So you have genomic regions which may be amplified or deleted, and you might want also to correlate those CNVs on the DNA with differential expression of the genes present in those regions. This is another view of what is meaningful in cancer genomics. Here we are speaking about structural variants, like for instance, as depicted here in this scheme, genomic fusions. So, for example, two genes that should not be together linearly on the genome, that instead are fused, so leading to a novel gene, so to say. And of course, this can have a very deleterious effect, leading to fusion transcripts with adverse effect and cancerogenesis. And finally, another way of looking at genomic alterations on DNA is searching for known mutations, of course, but as well as novel mutations, and hopefully trying to link those genomic point mutations on the DNA to variations in the transcripts, and of course functional and dysregulated transcriptional output. So there are many ways of getting hands to cancer samples. Of course, generally speaking, cancer samples are what we call complex and challenging samples. So, for instance, most of the cancers make use of tissue biopsy, which is a very invasive way of getting samples. And tissue biopsies mostly are getting fixed, those so-called FFP samples. And of course, this leads to DNA degradation, chemical modification of the DNA. And this is an example of a challenging DNA sample, as we say. But also, quite recently, the clinics is really going towards the use of non-invasive ways of getting cancer biopsies. You are mostly familiar with these term liquid biopsies, and they have proven useful for diagnostics as well as therapeutics. And what you are looking in these liquid biopsies from cancer patients mainly are circulating cells, so those so-called circulating tumor cells, CTCs, but you may want also to look at immune cells relating to immune therapy of cancers. So this is for the cellular part of the liquid biopsy component, but you may want as well to look at circulating nucleic acids, which are very diverse, so to say. So, like for example, cell-free DNA, abbreviated cfDNA, or circulating tumor DNA, so DNA released from circulating cancer cells, abbreviated here as ctDNA. So those CTCs and cell-free DNA that you obtain from liquid biopsies are typically very complex and challenging samples. So challenging in terms of quantities, of course, because you found there as quite rare events in the blood or plasma of patients, and of course they are also quite complex samples per definition, because, for example, cell-free DNA is a very small DNA species of roughly 170 base pair. So as you see here, this is just to summarize how circulating DNA can be found in the bloodstream, so released from apoptosis of tumor cells, or by necrosis, or it can be also secreted, and this is exactly what you aim for. So drawing some blood of patients, and either looking at so the cells which are circulating in the blood, or directly to the cell-free DNA. Okay, so this is getting a lot more attention, of course, in the cancer field, just because there's a need of non-invasive sampling of cancer samples, and so it's a very cost-effective way of getting samples from patients, and it has also the very strong advantage to allow for diagnosis of cancer, characterization of cancer, as well as monitoring disease progression and therapy. So what Takara Bio can offer for cancer genomics? Well, we offer a lot of different products which are currently recognized as industry gold standard for low input, complex, and single cell DNA-seq. So I just highlighted few of the samples or DNA types that we are specialized in. So for example, single cells, as we just discussed, circulating tumor cells, but also DNA coming from FFPE, DNA coming from plasma, cell-free DNA, circulating tumor DNA. And this is now achievable by our range of products, SMARTer, NGS, DNSeq, by leveraging two main technologies for DNSeq. So the throughplex one, which will be the first I will introduce you, which is mainly dealing with DNA per se, so like FFP samples and cell-free DNA, and also Picoplex, which is a single cell DNSeq technology. So all those two technologies are really recognized to be very sensitive, very robust, and reproducible, and this is precisely what you want to achieve in cancer genomics. On top of that, we will see that all the workflows are very easy and fast, which is a very good plus in cancer genomics. So let's start with the first range of solutions that Takaro Bio offers. So SMARTer throughplex, and we will see how it can benefit your research for CNV, SNV from plasma samples, putting the accent on cell-free DNA and circulating tumor DNA. So throughplex technology is a very easy technology. As you can see here from this workflow, this is a single tube workflow, only consisting of 3 very fast steps. So you will have to work basically 15 minutes with the sample, which is really fast. The advantage as well of having a single tube protocol is that just because we are dealing with complex samples which are present in low quantities, samples that are precious, of course, working in just a single tube allows you to retrieve the maximum of your sample, of course. So you see, very streamlined and easy protocol, and from your cancer samples to the sequencer, this all takes only 2 hours. So throughplex come in different flavors for cancer genomics, in 3 flavors. So throughplex DNSeq, this is a multi-purpose solution for cancer genomics. As you can see, it's one of the most sensitive products on the market. You can work with as low as 50 picogram of DNA, but I have to say that this one has not been really optimized for the complex samples we discussed previously in the introduction, maybe only just for FFP samples. When we are speaking about cell-free DNA, liquid biopsies, here we have a very optimized solution, which is called SMARTer throughplex plasma-seq. Really, it's been designed to deal with cell-free DNA, circulating tumor DNA, as you can read here, and also with a very low quantity of this kind of material. And finally, we have a solution for, let's say, when you are looking at rare mutations, rare alleles. So this means with a frequency of, let's say, roughly 1% and below, we offer this throughplex TAG-seq, which is taking advantage of unique molecular identifiers inserted during the adapter ligation step, and we will see later on how it can help in getting accurate results. So also here, as you can see, it is able to work with very minute amount of DNA. So this is the basic workflow of ThruPlex. As I told you, 3 main steps that you are mostly familiar with. So starting from your DNA sample from cancer patients, you will first do a repair of the ends, and then second step, adapter ligation. So all ThruPlex adapters are really unique in the sense that they are blocked at one extremity, the 5' end, which will prevent the formation of adapter dimers in your reaction, and this highly decreases, of course, the background and increases the amount of useful information. And then finally, the last step, the third one, will be incorporation of adapters and indexes and amplification of the library. So you can see 3 steps, very streamlined workflow, which is very beneficial for cancer genomics. So this was the workflow, general workflow for ThruPlex technology, which is the one found in Plasma-seq kit for cell-free DNA. And here I just put this scheme of the ThruPlex TAC-seq, where you see here on the right of the scheme, where we introduced the unique molecular identifiers in the adapters. So this is the yellowish little sequence here. And yeah, you can see that besides this, the workflow is shared with the previous one that I showed you. So talking about unique molecular identifiers, how can it help in detecting rare mutations? So when you're looking at rare mutation, of course, you want to avoid mainly bias in PCR amplification during library preparation, because, for example, this is on top the initial mutation that you're looking for in green, and during the process of library prep and amplification, sequencing PCR itself will introduce some little biases, of course, which are depicted here in blue and red. And taking advantage of the unique molecular identifiers, which in ThruPlex TAC-seq lead to more than 16 million different combinations possible. This will allow you then, after by demultiplexing, of picking the right mutation event on the DNA molecule. Okay, so this is very helpful for accurate rare mutation detection, and of course true variant calling. So I put the link below in green, where you can find more information on our website on how our unique molecular identifiers work and benefit rare mutation detection. Very important in cancer genomics, you may want also, so beside whole genome sequencing, you may want also to look at selected targets of interest in your workflow, and you can use, for example, target enrichment platforms, which are very well known, like for example Agilent SureSelect, X-gene lockdown probes, Roche Nimble gene libraries, and so on and so forth. You may want to perform exome sequencing as well. So all through-plex plasma-seq and TAC-seq have been validated with the target enrichment platforms that are shown here. We provide also protocols for use of through-plex along these target enrichment systems. And of course, we have many references documenting, so for plasma-seq and TAC-seq, the use of the kits along with exome panel for in cancer genomics. Okay, so I put also here in green the link on our website where you can find more information and all the dedicated protocols for target enrichment used with our through-plex kits. So I told you we have a very successful history of use of through-plex, plasma-seq, DNA-seq for cell-free DNA, liquid biopsies from plasma samples. And this is just a very recent example published last year, where this group looked at bladder cancer from and then looked at plasma samples from bladder cancer and looked at circulating DNA, and they were able, of course, to correlate the circulating tumor DNA in these samples with the clinical outcome of the patient. So this is one very important and useful application of NGS in cancer in liquid biopsies. So linking circulating DNA and monitoring progression of disease and therapy, and hopefully gaining insight into the outcome for the patient. This is also precisely what is referring to this very seminal paper published in Nature, where here this group from Cambridge looked at DNA present in the plasma of several and different types of cancers, and I reported this this paper just because they, on top of through-plex sequencing, they used whole exome sequencing. So this is also a further proof of combination of liquid biopsy samples, through-plex success in getting libraries from circulating tumor DNA, and target enrichment whole exome sequencing. Of course, I told you we have a long successful story for through-plex in looking at cell-free DNA, circulating tumor DNA from plasma sample of cancer patients. I just reported few very recent publications on these topics, and also this link in green to our website, where you will find, of course, a lot more references for successful use of through-plex in liquid biopsies of cancer patients. All right, so to wrap up this first part, so we saw that through-plex plasma-seq and through-plex TAC-seq are very sensitive solutions and really adapted for looking at cell-free DNA, circulating tumor DNA, starting as low as 1 nanogram of material, which is honestly very low. A very important features of both kits is that they both share a very fast simple, and single tube workflow. So really, this means that you will be able to retrieve the maximum of your samples, avoiding contamination and loss of material. And basically, because it's fast, you will be able to, of course, process more samples in less time. Very important feature is the compatibility and reference use of those kits with several major target enrichment platforms. And we really do think that the incorporation of unique molecular identifiers is really a big plus for accurate detection of rare mutations. I wanted also to mention here that throughplex chemistry is automated on this smarter Apollo system that we propose. I put the link here in grid. So this is a system which automates library preparation with throughplex. And just as final indications here, those are the various formats which are available for throughplex plasma-seq and TAC-seq. So 24, 48, 96, and 480 different reactions. So second chemistries that Takara Bio offers for cancer genomics is Picoplex. So we have launched 1 month ago this very innovative solution called Picoplex Gold single cell DNA-seq. So as from the name, it's meant for DNA-seq from single cells. And you will see in a second that it's perfectly adapted for combined CNV and SNV analysis from single cells. So looking at the workflow, this is something that you already seen in the previous slides. You may be familiar with now. This is the fast and easy workflow of Picoplex Gold. So here we only have 4 steps. Let's say 30 minutes, sorry, of hands-on time. So this is not so much of work, I have to say. And from single cell to the sequencer, it only requires 3 hours. And indeed, currently on the market, this is the fastest single cell DNA-seq solution. As you can see here, I reported some other solutions available, and you can see how stands Picoplex Gold. So it's very, very fast and convenient to use. So what is Picoplex Gold doing? How does it work? Well, this is summarized here with this cartoon. So starting from your single cell, so which can be circulating tumor cells or whatever circulating cell that you will retrieve from liquid biopsy from a patient. You will first use random primers that will extend the DNA molecules. Then you will have denaturation and repriming also with these random primers, extension, and all the sample will go through different circles of this reaction to perform, as I just said before, whole genome amplification. So in the 4th step here, amplification, the final incorporation of adapters and indexes for Illumina sequencing will be done. So this workflow is really leading to very high fidelity amplification of the DNA from the single cells. And yeah, I reported here on the right some advantages of this chemistry for single cell whole genome amplification. Also here, we have a very long-standing experience in the cancer field for picoplex chemistry. Those are only 3 references, but we have a lot more documented how picoplex is really the most accurate and reproducible chemistry for CNV detection from single cells. You could read that, of course, a lot of references put the accent on comparison of picoplex and other ways of doing whole genome amplification in single cells. And yeah, the consensus currently is pointing to, yeah, picoplex being, as from the title of the slide, being the most accurate and reproducible chemistry for CNV detection. Very interestingly, picoplex has been used quite recently, so in 2015, for this very innovative technology called GNT-seq. So GNT-seq stands for genome and transcriptome sequencing. So from single cells, in this case, circulating tumor cells, or they can be single cancer cells. And what is very important here is looking at both CNV and SNV from these cells, and of course trying to relate the genomic alterations to the transcriptomic alterations from those single cells. So in this very seminal paper in Nature Methods, picoplex chemistry was successfully used for this purpose. And now I will show you some very important data on the new picoplex gold single cell product that we very recently launched. So as we just saw, picoplex chemistry is really potent for CNV detection, and this has to relate somehow to a highly uniform and reproducible genome coverage achieved by picoplex. So what we did is that we compared picoplex gold with another chemistry, which is one of the leading one as well on the market. This is MDA, so multiple displacement amplification. And as you can see on the top panel, those are genomic examples, okay, loci. You can see that picoplex gold show really even distribution of the reads, whereas MDA somehow show regions which are not covered. So this is seen directly taking DNA as input, but very importantly, this is also seen this lack of coverage with MDA and good performance with picoplex gold from single cells. So the bottom panels here, each line is a single cell. So you can see that also at the single cell level, picoplex gold performs really well in terms of uniformity and reproducibility of coverage. And yeah, maybe it's small for you to see, but number 1 and number 2 represent here 2 different single cells. So you can see how consistent the genomic coverage is with picoplex gold. So this is another way of looking at how reproducibility is ensured by picoplex gold. So here looking genome-wide at genome coverage, so in different bin sizes, so relatively small ones and bigger ones here. So you would expect, of course, a perfect correlation between both single cells, one on the x-axis and and 2, sorry, on the y-axis. by having this straight line with a coefficient of correlation of 1. You can see that Picoplex Gold almost achieved perfection in terms of reproducibility, which is definitely not the case with MDA. So here, a very important aspect when looking at single cells and doing whole genome amplification from single cells is, of course, the highest reproducibility that you need for your single cell analysis. And this is achieved by Picoplex Gold. Finally, so we had a look at CNV-related genome coverage, uniformity, reproducibility, but another very important aspect, as we discussed in the introduction, is also SNV detection. So looking at point mutations. So I would just give you first the color key here on the left. So in blue, we are looking at Picoplex Gold, our newest kit. In gray, this is the MDA chemistry from QIAGEN. Okay. And here, the first graph depicts how many SNVs Picoplex Gold is capable of detecting from single cells. So in blue here, compared to MDA in gray on the left. So you can see that we are able from single cells to detect 3 times more SNVs using Picoplex Gold. And to give you an idea of the numbers of SNVs detected, because it might be small for you, we are above 300,000 of SNVs detected by Picoplex Gold, which is definitely a lot, even from single cells. So of course, high numbers of SNV detection is good, of course, but you need accuracy in your data, most of all when you are looking at point mutations. So this is what we looked on the bottom graphs. So the first one is so the allele dropout frequency. So is Picoplex Gold good as detecting the mutations that you need to detect? Do you miss some of the information or not? So you want to keep this frequency of allele dropout the lowest possible. And this is what you can see here in blue with Picoplex Gold, which has very low allele dropout frequency respect to MDA. So this also at the single cell level. Another way of looking at accuracy and robustness of your detection of SNVs is looking at false positives in your data. So in that case, we evaluated allele drop-in frequency, and you can see that it's really maintained at a very low level. So here we are reaching 0.05% of alleles dropping. So this is even performing better than MDA here in gray. So really to summarize the high performance of Picoplex Gold that we already saw for CNV, these graphs really points to a very high standard, and mostly we think gold standard for SNV detection in single cells, in single cancer cells, circulating tumor cells. So first, in terms of high rate detection of SNV, and of course you can trust this data because allele dropout frequency and drop-in frequencies are very low. So those are data that you can trust. So to wrap up this part on DNA-seq in single cells, showing Picoplex Gold as our leading solution. So with Picoplex Gold, now we propose really a best-in-class system for DNA-seq in single cells. So I really wanted to highlight those three features here: sensitivity, reproducibility, and accuracy. So this is really what you are aiming for in single-cell DNA-seq, mostly when you are, of course, when you are looking at CNVs, but mostly when you are looking at SNVs. And Picoplex Gold performs extremely well for both of these analyses. On top of that, I think it's very important to stress again that the whole process is very fast and simple. So the fastest workflow right now on the market. And this is really a solution, Picoplex Gold, which is outperforming by far. Let's say one of the method which is still used a lot for SNVs, which is MDA. So Picoplex Gold definitely outperforms MDA, as I showed you. This is a single cell solution, so we have those three formats available: 24 reactions, 96, and 384 reactions. You will find on our website a lot more information on ThroughPlex Plasma-seq, ThroughPlex TAC-seq, and Picoplex Gold solutions. So I reported here the link to our website to our DNA sequencing solutions, and also the links here to the products we discussed today. And I wanted also to mention that we recently added to our website a very dedicated section, which is, of course, dedicated to cancer research. You will find it here at this link, and you will find also information on single cancer cell genomics, as well as cancer genomics and epigenomics. And that's good science.